introduction. So our next speaker is Catrina from University of Pennsylvania, and she has been doing work looking into visual processing and looking in non-human primates single neuronal firing and try to relate both that to perception and memory. And of course, this is a topic very relevant to what we are discussing here today. So we are very much looking forward to your presentation.
Thank you. So thank you everybody for listening to me speak. To start my presentation, because this workshop is focused on the intersection between artificial neural networks and cognitive neuroscience, I thought that I'd start by laying out how I see those two things coming together and where I think that my work fits into that. So it goes without saying to this audience that artificial neural networks have been a powerful tool for modeling the visual system. So not so long ago, in 2005, a group of researchers at the forefront of modeling early visual cortex got together and wrote a paper about the state of the art in modeling the visual system. And in it, they wrote that an adequate model of visual responses should predict responses to arbitrary stimuli, so that includes natural stimuli, but that at the time, many of the standard models of early visual processing had not been held to that rigorous test. And I share this because within a decade, artificial neural networks were already being used to accurately predict visual responses, not only to arbitrary stimuli when presented, or to early visual cortex to arbitrary stimuli, but also throughout the ventral visual stream, including in areas that were once thought to be impossible or difficult to predict, like IT. In fact, artificial neural networks have gotten so good at this object recognition task that modern advances are, at least in this task, are focused on these small enhancements in performance or coherence with behavior and neural data. And while there's certainly a lot of important work still to be done in improving the correspondence between networks, brains, and behaviors in the object recognition task, there's no doubt that there are artificial neural network models that are good enough predictors of the geometry of neural responses in the object recognition task that we can start to leverage their predictive power to ask questions that go beyond just object recognition. And one such compelling set of questions aims to identify the neural correlates of cognitive processes like attention or memory, which is motivated in part by their applicability to neurological disorders and diseases. One of the most productive paths towards success in this endeavor has been to study these processes in the context of the visual system, for example, by studying visual memory or visual attention, where a strong foundational knowledge of visual processing aids in the disentangling of cognitive processes and visual signals. So we can go from asking questions like, what am I looking at, to questions like, how do I remember what I've seen? And this is part of the reason why I study visual recognition memory. And just as artificial neural networks have been a powerful tool in quantifying the once unquantifiable when it comes to object recognition, so too can they be a powerful tool in this realm of more cognitive questions. In my work, I leverage the predictive power of artificial neural networks in this way. So not necessarily taking them as models of the brain per se, but taking advantage of their predictive power to quantify and parameterize features of natural image space, which would otherwise be impossible to quantify. And specifically in sharing my research today, I hope to highlight two places where I see ANNs and cognitive neuroscience benefiting from each other. The first is in the ability of ANNs to parameterize natural image space to give us experimentalists new knobs to turn in studying cognitive processes like memory. And I hope that what I mean by that will be even more clear once I present my own work. But also in the use of ANNs as benchmarks to help us to identify the contributions of other brain processes to behavior. These are the big picture that I'm hoping that you'll take away from my presentation, and anything that you learn about my project along the way will just be icing on the cake. So I said that I study visual recognition memory. I think that this is a really promising research target, not only for all the reasons I just laid out about our ability to take advantage of computational tools in vision science, but also because humans are effortlessly very good at this kind of memory. So in the lab, subjects can remember thousands of images, each presented for only a short amount of time. And these memories are highly specific for image details. So for example, human subjects can really easily remember which of two very similar images, for example, the top or bottom row of images here, that they've seen previously. And so it's probably no surprise that visual memory has been the subject of many studies in psychology and neuroscience. Specifically, it's been extensively studied using paradigms in which subjects are shown a sequence of images and are asked to report when they recognize an image that they've seen before. And now decades of research now has used this kind of a paradigm to build up a really strong foundation in our understanding of the neural mechanisms that support visual recognition memory. But my work focuses on a feature of real-world vision and visual memory that's not captured by this kind of a paradigm. Specifically, by presenting images from many random categories in this way, these studies assume that every object is equally likely to appear. So for example, that we're just as likely to see a dog after we've seen a sunflower as we are to see a giraffe after we've seen a city skyline. However, we know that that's not true of the real world. If you go to the dog park, you're going to see lots of dogs of different shapes and sizes and colors. But if the next thing that you see is a giraffe, that's probably pretty surprising. And the same is true of cars on the highway, vegetables in a farmer's market, desks in a classroom. And we know that as we go about our day, that these kinds of changes in the statistics not only influence visual representations through mechanisms like sensory adaptation, but there's also been behavioral work showing that this has a meaningful impact on visual memory. So in this foundational paper, Zoya Belinsky and colleagues showed that while we're very good at remembering a given image when it's shown in a sequence with objects from many different object categories, which is what I've told you previously, if you take the exact same image and you now present it in a sequence that has only images from the same object category, they become much less easily remembered. So in this case, I'm using, for example, the category of dogs. And this paper did a really good job demonstrating behaviorally that changes in the statistics of a sequence influence visual memory, but the neural correlates of this effect have not been investigated. And I think that they're interesting for many reasons, but importantly, and one reason is because they are help us take a step toward understanding more naturalistic vision and visual memory processes, but also because understanding the neural correlates of these effects puts critical constraints on our understanding of where and how visual memories are formed and stored. So given these motivations, I've been working for a while on this project, aiming at identifying the neural correlates of contextual influences on visual memory. So to do so, I designed a task in which I systematically manipulate temporal context by showing blocks of images with different statistics, and then I record from visual and memory areas as a monkey performs the task. So the core task that we've trained the monkey to do is to view a sequence of images one at a time and to determine for each whether it's a novel image or a repeated image. The first kind of block that images could appear in are what I call random blocks, in which images come from many different categories, and that's color-coded in black. So this is like the classic visual memory experiment that I was talking about previously. The monkey is trained to make a saccade up or down for each image to indicate his choice. And the images appear exactly twice, once as novel and a second time as repeated. And the interval repetition is fixed at about 35 images, which corresponds to like a couple minutes delay. So this is the random block. This is the same kind of paradigm that's been used previously. So in order to investigate the impact of context, I've introduced this second type of block, which I call a categorical block, in which 80% of the images are from the same category, in this case, dogs. And I call these categorical images. I will color code them in blue throughout my presentation. And then the other 20% of images I pull from random categories, and I call these oddball images color coded in red, which is kind of like a random control that occurs within the categorical blocks. And the monkey views each image for a fixed 500 milliseconds before the image disappears from the screen and the monkey reports a decision. So this is the task that we've designed, but we want to make sure that we're capturing the full richness of these effects. Specifically, we know that memory for all images is not going to be equally impacted by putting them in the context of their category. So going back to that original paper that I was talking about, those researchers actually found that the amount by which an image is impacted by placing it in the context of its own category is actually really variable depending on the specific category that you're using. So for example, if you have a category like living rooms where the images are not necessarily visually very similar, you get really small effects of context on memory, versus in a category like lighthouses where the images are very visually similar, we get really strong impacts of context on memory. And so the takeaway here is that the homogeneity of an object category or a sequence of images, so the visual similarity, is modulating visual memory. And so we want a way to quantify that similarity so that we can investigate the impact of that on visual memory processes. So for my task, going back to our dogs example, I'm looking for a value that can help me distinguish a dog that's very similar to all the other dogs in a sequence from a dog that's very different from other dogs in the sequence. And without artificial neural networks, we do have a few options for how to do this. The first is we could use low level features of the images, like, for example, pixels, to quantify the similarity between images. However, that is not necessarily a good idea because we know that these kinds of features don't predict the perceptual similarity reported by human subjects of images very well. Or we can do behavioral judgment. So we can ask subjects to tell us for any two images how similar they think those images are. However, that's very expensive because we now need to get behavioral reports for thousands of images in our experiment. And it's also really hard to make that sequence specific. So we can get measures for any two images, but it's a little less clear how we would do that to say for this image in the context of, say, the last 10 or 15 arbitrary images that I've shown. And so that's why here is where that first point that I made about artificial neural networks starts to come into play in my own work. So by leveraging an artificial neural network, in this case, we use the network CoreNet to get noiseless estimates of the representations of each image at the highest level of the visual hierarchy, where we know that those features correspond to perceptual similarity reported by subjects. We can now get a measure for thousands of images that we suspect will be predictive of perceptual judgments. And we can do that in a sequence-dependent way for arbitrary sets of sequences. And we're doing that all essentially for free. So again, this is an example of that first point that I wanted to make about using artificial neural networks to put these like quantitative knobs that we as experimentalists can turn to study these processes. And the measure that I developed using this is what I call block similarity. So to compute this, I start by collecting the activation pattern across all 25,000 artificial units in the IT-like layer of CoreNet for each image in a block. And I take correlation as a measure of similarity. So I begin by correlating every pair of images in the block, every pair of activation patterns in the block. And that gives me a correlation matrix for each block. So for example, these two images are not very similar to each other. And so they should have a low correlation between their activation patterns. So block similarity is simply the average similarity between any one image and every other image in the block, which is just the mean across any column of our correlation matrix. So the result is that now instead of saying, okay, I have a dog that looks similar to other things or less similar to other things, I now have a number where the dogs that look most like other dogs in the block will tend to have a higher average correlation, and thus a higher block similarity, versus dogs that look very different will tend to have a lower average correlation and thus a lower block similarity. So I told you that because I used the activation patterns from CoreNet, I have an expectation that this is predictive of perceptual judgments. But we want to validate that the features that we've extracted are in fact relevant to visual memory. And to do this, we now look at the monkey's memory performance as a function of my new block similarity measure. So here I'm showing the proportion of trials that the monkey got correct as a function of block similarity. First, we see that for random and oddball images, they tend to have a low average similarity, which is shown by the shift to the left on this x-axis. That maps onto our expectation. We expect that random objects should on average be very dissimilar from each other. And we can also see that the monkey's memory performance is quite high, so with near perfect performance for these images. Categorical images, on the other hand, have a much broader distribution of block similarities, and they're on average higher than for random and oddball images. They're shifted to the right on that x-axis. And that again maps onto our intuition, because dogs should be more similar to other dogs than they are to random object categories. We can also see that the monkey's memory performance on average is lower for the categorical than the random and oddball images. This is recapitulating those human behavior results that I have represented to you a little bit ago. But we can also see now that the monkey's memory performance tends to drop off systematically as a function of block similarity, which gives us a really nice behavioral effect that we can now leverage or utilize to investigate the neural correlates of this behavior. So now I'm going to start talking a little bit about the neural data that I simultaneously collected. I collected neural data in visual cortex, specifically in IT, and in medial temporal lobe, specifically the CA3 and dentate gyrus subregions of the hippocampus. And remember, I'm recording this while the monkey is performing the memory task. So I have simultaneous behavior and neural measures. You may think of IT as a visual region and hippocampus as a memory region, but both of these regions have been implicated in visual memory, and hippocampus is thought to contribute to higher level features of memory like context. But I'm going to tell you in a little bit about how our lab thinks that those two processes are happening in these brain regions. I simultaneously recorded dozens of cells across 40 sequential sessions, 13 in IT and 27 in hippocampus, to get matched numbers of visually selective units in each population. And then I align those images in a manner consistent with methods demonstrated to be effective in past data sets from the labs that preserves the block similarity of images. So the main takeaway from this is that I end up with neural populations where I have the responses of 535 units to 47 oddball, 80 random, and 144 categorical images in IT and hippocampus, where I know the block similarity of each image. But before I can unpack my neural results, I'm going to share how we think that visual memories are represented and stored in these brain regions. To gain an intuition for population responses, we often think of population responses as a vector in high dimensional space, where each dimension is defined by the response of a single neuron. So images that have similar representations and thus more overlap in their responses will lie closer in this space, while dissimilar images, which should have less overlapping representations, will lie further in this space. Thus, visual identity is coded in the distances between these vectors. Now, we're interested in visual memory, and when we see something and we get this sense that we've seen it before, that signal has to manifest itself somewhere, and the question is where. And our lab has published quite a bit of work now suggesting that this is through a phenomenon known as repetition suppression. So repetition suppression describes the fact that when an image is repeated, it tends to evoke a weaker population response than on its novel presentation. So in our geometric space, we can visualize this as a shortening of the response vector. And now you can see, so the idea is that the memory signal manifests itself through response magnitude, and that is orthogonal to the identity signal, which is manifesting itself through the angular distances between these responses. And this is how we think that these brain regions multiplex visual and memory information through this orthogonal coding scheme. To show you that what repetition suppression looks like in these brain regions, you can see I found robust repetition suppression in both IT and hippocampus. Here I'm showing you the reduction in the response to repeated relative to novel images as a function of time after stimulus presentation for my IT population in purple and my hippocampal population in green. The existence of repetition suppression is not disputed, and it's been documented throughout many brain regions. But the interesting question is whether or not that is the signal that predicts memory. And the lab has shown in previous publications with IT and hippocampal data that repetition suppression is in fact a memory signal that predicts behavioral performance. And specifically, we do this by using a family of linear decoders that all rely on the reduction in response that can successfully decode neural activity to faithfully predict a variety of visual memory behaviors. The simplest of these decoders is called the spike count classifier, which I've popped up on our geometric space here. It just counts the spikes for novel presentations of images, and then the spikes for repeated presentations of images, places a decision boundary between those two distributions, and now any new image that evokes a response that's greater than the decision boundary will be classified as novel, and less than will be classified as repeated. And of course, all of these classifier analyses are fully cross-validated to avoid overfitting to the data. And we've published work showing that this is true for IT, and that IT is sufficient to account for various features of memory, like, for example, forgetting over time, or invariance to visual features like contrast. But what about a more cognitive variable like context? There's a lot of work in cognitive neuroscience speculating on important roles that the hippocampus might play when it comes to these kinds of variables. And I'm going to talk actually about a specific one of these hypotheses in a little bit. But the question one of the questions that my project is aimed to answer is IT sufficient to account for the contextual effects on visual memory, or is the medial temporal lobe also engaged in important computations? And to answer this question, I'm going to give you two angles that we took to answer this question. The first, we can look at this with respect to the memory representations by applying that classifier analysis that I just described to my data, which once again, just an important thing here is we can interpret the result of that classifier analysis as a readout of our ability to relate repetition suppression to visual memory. So here I'm just showing you the same behavioral data that I've already shown you. The dots are the monkey's memory performance. And just as a reminder, the key behavioral effect is that the monkey's memory performance drops off as a function of block similarity. So now I'm showing the performance of our memory classifier when it's trained on IT data, and it's been rescaled to match the levels of the monkey's behavioral performance. And the key takeaway here is we can see that the classifier that's trained on IT data does show a drop-off in performance as a function of block similarity that matches the kinds of trends that we see in the behavioral data. So another way of putting that is that repetition suppression in IT is predictive of the monkey's memory performance. And that already gives an answer to our question, which is that, yes, actually, the contextual effects on visual memory are present as early as visual cortex, specifically IT. But we can still ask the question, so then what might the medial temporal lobe be doing? Can we still decode the monkey's memory performance from that neural data? And so now I'm showing the performance of the memory classifier when trained on the hippocampal data as a dotted line, again, rescaled to match the levels of the monkey's behavioral performance. And like the classifier trained on the IT data, it shows on average a drop in performance compared to the random and oddball images. So that does recapitulate that average performance that we saw from the humans. However, unlike IT, it doesn't show the systematic drop-off in memory as a function of block similarity that we see in the monkey's behavior. And while I'm still doing some work to really tease apart exactly why we see this discrepancy and like what's going on here, I want to pause and I just want to highlight that without the block similarity measure, which again was only possible because of the way that we leveraged the artificial neural networks, we wouldn't be able to tease apart these differences between hippocampus and IT, because you can see that hippocampus is on average showing a decrease in the performance for these images relative to the random and oddball images. And it's not until we introduce this quantitative knob that we're turning called block similarity that we can begin to tease apart what these two populations are doing. Okay, so as I said, this is evidence that visual memory representations as early as IT capture contextual effects on visual memory. But now we've talked about the memory representations. What about those identity representations? Might context be influencing the identity representations differently in these brain regions? And luckily, the literature points to really clear hypotheses about what these brain regions are contributing here. The most famous of these is what's called the hippocampal pattern separation hypothesis. The idea here is that a core static computation of the hippocampus is to take visual representations that are correlated or overlapping in visual cortex and to make them more decorrelated to separate them before memory storage. And the idea here is that memory is a noisy process. And so by decorrelating representations, we're preventing the risk of collapse of the representations, which would cause confusions in memory. So this, as I said, this is a pretty famous hypothesis about what hippocampus might be contributing. However, we again have an idea of something that cortex might be contributing. And specifically, vision science suggests that sensory adaptation in visual cortex decorrelates visual representations in contexts where you see lots of similar images. So it's like an adaptive process that we expect might be at play in categorical blocks where the monkey is being presented with lots of very similar images. And so sensory adaptation might be at play decorrelating those representations. And just to point out, these are not mutually exclusive hypotheses. We could see evidence for one or both of these processes at play. So the question is, what do we see? And here's where I'm going to illustrate that second of those points, being the importance of the artificial neural network as a benchmark for comparing brains and like real squishy brains and the models of them. The challenge here is that I had a single exposure task. So I only have measures of the representations of these images in one of the blocks, so either random or categorical. And to test my cortical pattern separation hypothesis, I need to compare representations in categorical and random images. So to get around this problem, we use CORNET as a measure or a prediction of the representations of images in a random block. So first, I'm just going to show you that that's a fair thing to do. So when we look at the representations in random blocks, specifically those block similarity measures, we can see that the network, being CORNET, is actually a pretty good predictor of the average similarity that we see between these representations in IT. However, now the interesting question is what happens when we go to categorical blocks where the network predicts that things should be more correlated, but we think that there might be adaptive brain mechanisms at play that are decorrelating these representations. And that's exactly what we see. So you can see that the representations in IT tend to be more decorrelated than predicted by the network. And we take this as evidence for this adaptive cortical pattern separation hypothesis. So this is interesting. So I've just showed you that context once again is doing something meaningful, it seems, at the level of visual cortex. How about in hippocampus? This is a simple comparison now between IT and hippocampus, the similarity of the representations there. And we can see that once again in both blocks now, because again, this is a static computation we think that hippocampus is engaged in, the representations do tend to be decorrelated relative to their representations in sensory cortex. So we take this as evidence for, or at least consistent with, our hippocampal pattern separation hypothesis. And so the takeaway here is that we have context is acting on the visual representations as well as the representations in the medial temporal lobe, and that we're able to leverage the predictive power of the artificial neural network to distinguish, or not to distinguish, but to highlight the functional consequences of at least some adaptive brain processes that are happening in categorical blocks that are not captured by this state-of-the-art artificial neural network. Okay, so stepping back in terms of my research that I have shared, some of the key takeaways from what I've shared today is first, hopefully you can appreciate that context matters. I've also showed you that we can predict image-specific changes in a monkey's visual memory performance, and I hope that the takeaway from that is that artificial neural networks were key in developing this quantitative knob called block similarity that really helped us to tease apart the computations that are occurring in the brain. I also hope that you'll walk away from this presentation with a sense of don't underestimate the role of cortex when it comes to visual recognition memory. I think we often think of hippocampus as being like a memory region, but a lot of my work has pointed to important heavy lifting that cortex is doing. And finally, I pointed to a place where cortical representations and artificial neural networks are in disagreement. There's something that the brain's doing that artificial neural networks are not. And so that's a potentially interesting point for, say, a computer scientist or someone interested in using artificial neural networks as actual models of the brain might step in to try and capture. And to finally bring it all home, this is the slide that I started my talk with today. And I hope that after seeing my work, you can appreciate these 2 points at the intersection between artificial neural networks and cognitive neuroscience. And I'm really excited to continue working at these points of intersection. So I just want to say thank you to my co-authors and to other members of their lab for their help throughout this project, and I'll take some questions.
Thank you very much. Very fascinating work. Questions, Mark?
Very nice work. Can you comment on the reset time for the repetition suppression? Because in some of the older familiarity memory work, they seem to say that it lasts forever. Once you've seen it, you always will suppress. But I can imagine that in the task, you can give the monkey the task to reset and start afresh, so to say. Do you see effects of that?
Yeah, so I think you're asking like the longer time that you have between the first and second time that you see an image, does that change repetition suppression? Yeah. So I actually don't have a figure for that, but one of the first papers in the space that the lab published was looking at the effect of N-back. So that's how long is there between the first and second time that you see an image. And so I told you I set that at 35 images, which means that I expect to see about the same amount of repetition suppression. But what they found is that it drops off as a function of N-back. So if you show them right after each other, you can see a reduction as big as like 15% between the novel and repeated images. However, when you get to later numbers like 64 images between the first and second time that you see it, in some monkeys, the repetition suppression goes away entirely. And so this is definitely, we're looking at like a short-term recognition memory signal in terms of like what happens to convert that into long-term memory. I don't have anything concrete to say about that, but I'm definitely in that range where we see that this signal is relevant.
Thank you.
Yeah. So Gali has a question.
Thank you for the presentation. I have a question. So you showed that the block similarity is more similar to the IT activity rather than the hippocampus. But if I remember correctly, your block similarity is estimated from the ANN, but from the layer of the IT layer. So is it bias towards the similarity towards the IT rather than hippocampus. And another question is, here you classify the context as the same category, right? Images from the same category. But usually for hippocampus, the context information is more like kind of episodic memory, like high level context. So is this also possible? Because the definition here of context is not really the classic definition in hippocampus.
Yeah, okay, I think I caught both of those questions, and I'll try to remember and answer both of them. So the first was a question about I'm defining context or my block similarity using the IT-like layer of a network, and so am I biasing toward a better match to IT than to hippocampus? To that I say, like, that is possible, right? But I think the thing that I can kind of hang my hat on is the fact that regardless, this is something that is predictive of the monkey's visual memory performance. And so there might be other axes along which the monkey's memory is also modulated that I'm not capturing with this measure. And maybe that's what hippocampus is doing. I'm definitely investigating that kind of question. But I think that there's no doubt that I am capturing something that is influencing the monkey's memory. And that's all that I can really say anything about. The second question, can you remind me what the second question was?
It's for the context. So here you define the context as the same pictures from one same category. But usually when you talk about hippocampus, the context is more like high level. Like for example, in episodic memory, you have different categories of information involved, right?
Yeah, so I totally hear you there. And I think there's a lot of evidence that even brain regions like prefrontal cortex might start to come into play for those kinds of higher level categories. I think that would be interesting to investigate. Obviously, I'm not looking at that level. I've always been really interested in things like what happens when you change like the statistical regularities of the sequence, not just at the level of category, but you say like, I expect to see certain sets of images, those kinds of things. I am very open to the possibility that those things are encoded in different ways in different places. But I still think that this idea of visual similarity is really important and worth investigating.
So I have another question related to sharpening, right? So I fully get the sort of context of your full story, repetition, suppression, and less memory. However, one could also argue in natural situations, you might be saccading and then sort of revisiting the same object. And when doing that, you get more certainty about that object, sort of accumulation of evidence. And that at first seems to be at odds with repetition suppression. However, then people have also talked about sharpening. So it could be that fewer neurons partake in a second and third time when you see the representations, but you have a more precise representation. Is your framework also consistent with incorporating sharpening?
Yeah, so I mean, I think the sharpening is what would come in in these visual identity representations, right? Because we're talking about like changing the tuning curves of the individual neurons. Our lab has done some unpublished work trying to look for evidence of this kind of a sharpening hypothesis, and we've found it pretty hard to find any evidence for something like that, but that doesn't mean that it's not at play. And I do think that those kinds of things would show up in these identity analyses that I'm doing, where I'm looking at differences in the representations.
Yeah, thanks. So I see Catrina has a question.
Yes, actually. Hi, Catrina. I'm just curious. So I know there was this trend with CNNs, or it's been there for a long time with brain score and CNNs and the visual system, but it seems that now more state-of-the-art networks actually become less similar to the visual system. And I've also been told that what becomes more and more similar are large language models, which would have recurrencies, and they would also be able to learn this context variable. So I'm just curious if you would think that a large language model would be more similar, for example, to the hippocampus, or if you like have any thoughts on how this might using such a model would sort of change your results?
Yeah, I mean, so to be honest, I don't know a lot about the details of large language models and how they're implemented, but the part of what you said that does resonate is one thing that I'm looking at is recurrent neural networks. And so what happens when you build that kind of, or allow for that kind of dynamics, and you can track context, can you then capture these effects better? And I completely agree with you. Maybe this discrepancy that I have between hippocampus and behavior can be accounted for if I can model those dynamics in something like a recurrent neural network.
More questions? So maybe on what the discussion we're just having now, I mean, can you also think about, is it possible to sort of modify CoreNet such that it takes the dynamics into account so it can do the repetition suppression?
Yeah, so there has been some work doing things like this. I think it's a little bit harder to get the image specific repetition suppression built in, but there's one really nice paper that just adds adaptation into a feed forward network. I think specifically they used AlexNet, and then you can ask the question, is feed forward adaptation sufficient to account for these different kinds of behavioral effects? And so that is something that I've not yet done, but I would really like to look at, because that helps you to tease apart then what of the feed forward process, just adding adaptation, can account for these kinds of effects. In general, it's really hard to capture the image specific repetition suppression across, because it's so robust across so many intervening images. But perhaps some of these contextual effects will be more easily captured.
Okay, thanks. More questions? Okay, then thanks a lot for a very interesting talk.
Thank you.